For performing phylogenetic analysis, we have to do it in 5 different stages. First stage is related to sequence acquisition processing. Sequence acquisition से क्या मुराद है? It simply means to attain some sequence from a stored place. इस मॉड्यूल में मैं दो चीज़ों पर फोकस करूंगा कि व्हाट आर डिफरेंट डेटाबेसेस अवेलेबल फॉर बायोलॉजिकल डाटा विच वुड बी अ जेनेरिक थिंग, एंड देन स्पेसिफिकली how these information is important or relevant for phylogenetic information. So there is a standard institute which is National Center of Biotechnology Information owned by USA, which is basically a landmark place for sharing information related to biological data. So what does it do is that it has list of established publicly available databases of biological data. Then they provide information for research and development for biological data. Then they have a list of developing softwares to handle those biological data. And then finally, it is responsible for distributing or disseminating information across different organizations which are working on biological data. अब बायोलॉजिकल डाटा कितनी टाइप का होता है और हाउ मेनी टाइप्स ऑफ डीबीज विच आर रिलेटेड टू बायोलॉजिकल डाटा आर अवेलेबल अक्रॉस द वर्ल्ड. Broadly speaking, there are 5 different categories of DBs. Number 1 is sequence databases. What are sequence databases? वो databases जिनमें हमारे पास मैक्रो मॉलिक्यूल्स की बेसिक इंफॉर्मेशन मौजूद है, जैसे डीएनए, आरएनए और प्रोटीन स्ट्रक्चर्स के जो बेसिक स्ट्रक्चर हैं या सीक्वेंसेस हैं उनको स्टोर किया गया है. Then we have bibliographic databases. Bibliography means literature related studies, means that what we have information which is related to research community. Third one is clinical databases. Clinical databases are those databases which are made by humans for patients particularly. Fourth one is integrated databases. Integrated databases are combination of different databases. For example, secondary databases can be part of this one, which are derived from primary or sequence databases. And finally, we have structural databases. Structural databases are those databases where we have information in different formats, such as, for example, for proteins, we have that information in more than one coordinate, might be 2D or 3D coordinates. आइए इनकी थोड़ी डिटेल में चलते हैं कि वो कैसे होंगी और वो क्या इंफॉर्मेशन अटेन रखते हैं. इस मॉड्यूल में मैं फोकस ज़्यादा ये करूँगा कि आई विल प्रोवाइड लिस्ट ऑफ दोज डीबीज विच आर फ्रीली अवेलेबल और कैन बी सर्च फ्रॉम इंटरनेट. So for sequence databases, we have these 3 major sequences for DNA type information. For example, we have GenBank, then we have DDBJ, which is Data Bank of Japan, and third one is our EMBL, European Molecular Biology Laboratory database, and then we have SysProt, which is for protein database owned by Switzerland laboratory. Similarly, for bibliography DBs, they are, as I said, they are used for searching reference material or literature based materials. And what happens is we have a PubMed search engine from NCBI, which is basically a search engine which brings information from a Medline corpus. Medline corpus is basically collection of documents which are related to biology, and this PubMed search engine, it usually or at current times it can have information attained from roughly 12 millions of documents which are kept by Medline corpus. Similarly, we have Web of Sciences, which is free for academia, and we just have to make a username and password, and then we can attain information from Web of Sciences. Similarly, we have a PubCrawler, which is also free, and then it contains information about journals, and then these journals are basically related to information about biological data. And what we happen is they whenever the good thing is we just give our query, and then whenever they find result they will give us back. So moving on, clinical databases, there is a human gene mutation DB which is owned by UK, and then what happens is it registers known mutations in the human genome and then the different diseases caused by that genome. Similarly, we have DBSNP, which is from USA, and it is largest DB for single nucleotide polymorphisms, and it also compares its results with other SNP databases. Similarly, we have integrated databases. So what happens is this is basically amalgam or mixture of different other DBs and then making one unique DB. So we have gene cards, which is basically keeping information about complete gene sequences, and it would be very thorough information, and it has also links with other informations or other DBs also. So moving on, we have IntroPro, which is basically integration of individual protein sequences such as prints, PROSIT, SMART, and so on. So what will happen whenever we give an information to search a protein, it will search that given protein sequence against all the other available protein sequences which are in these different protein databases. So finally, we have structured databases. One example of this structured database can be the tertiary based databases which are useful for proteins. So here we have protein data bank, which basically has information of protein data structures in the form of 3D coordinates, and it is very useful for showing the humans or for so why it is important, because humans can find information of individual items which are shown as a 3D structure, and it will be very visually appealing. Similarly, finally, we have EMSDB or SCOPE and CATH. Basically, all these are related to protein structures. For example, this SCOPE, structure classification of proteins. So what it does, it just instead of just showing protein structures, it also provides annotation. Annotation means some tagging or some name which are attached with the protein structure. And finally, for CATH, we have classification, architecture, topology, and homology of protein structures. So it tells us what is the architecture, what is structure, what is topology, means what is the broad category of those protein structures, and finally what are homology of those protein structures, what are from the same common ancestor information for a common protein.